Welcome to section 12 of genetics. In this section, we'll be discussing microdeletion syndromes. Let's get started. Microdeletion syndromes are a group of disorders caused by a deletion of a segment of a chromosome that spans multiple genes but is less than 5 million base pairs in length. In other words, it's not a small mutation in a single gene like sickle cell disease, but on the other hand, it doesn't involve an entire chromosome like Turner syndrome. So I like to think of it as a medium-sized deletion somewhere in between the two extremes. Microdeletions are usually caused by sporadic recombination errors that occur during meiosis. There are 3 microdeletion syndromes you need to know for step 1, including Cri du Chat, Williams syndrome, and DeGeorge syndrome. This is a figure of meiosis, which can be found in section 2 of genetics. Notice that recombination occurs here between 2 homologous chromosomes. It's during this process that microdeletions occur. This is a table of the microdeletion syndromes, which can be found in section 12 of genetics. Cri du Chat syndrome is caused by deletion of a segment of the short arm of chromosome 5. If you recall that Cri du Chat is French and means cry of the cat, then you'll be able to remember the prominent feature in this disorder is a high-pitched cry at birth that sounds like a meowing cat, so it's similar to a cat. Other findings include intellectual disability, microcephaly, congenital heart disease including VSDs, and epicanthal folds. This is an image of a boy with Cri du Chat, which can be found in section 12 of genetics. Williams syndrome is caused by deletion of a segment of the long arm of chromosome 7, which includes the elastin gene. Some of the symptoms can be remembered with the mnemonic Will Ferrell in the Elf is very friendly. These individuals have an elfin appearance and are extremely friendly. Other features include intellectual disability, well-developed verbal skills, congenital heart disease, renal artery stenosis, and hypercalcemia, which is due to increased vitamin D sensitivity. This is an image of several individuals with Williams syndrome. Notice that they generally appear very happy. They also tend to have broad foreheads, full cheeks, and short noses, giving them the characteristic elfin appearance. DeGeorge syndrome has many different names, including 22q11 deletion syndrome and velocardiofacial syndrome, but it's most commonly called DeGeorge syndrome. This is a microdeletion disorder caused by a deletion of a segment of the long arm of chromosome 22. More specifically, it's a deletion of 22q11. This results in aberrant development of the 3rd and 4th pharyngeal pouches. These are responsible for the development of many structures, including the thymus, the parathyroid gland, pharynx, and the upper portion of the heart. The symptoms can be remembered with the mnemonic Catch-22. C for cleft palate, A for abnormal facial features. It's high yield to know that one of the facial features is hypertelorism. Hypertelorism just means there is a wide space between both eyes. T stands for thymic aplasia. Because the thymus is associated with T cells, this results in a T cell deficiency, so these patients are immunodeficient. This also results in an absent thymic shadow on X-ray. C stands for cardiac defects. You can see I've shown many possible cardiac defects in parentheses here. H stands for hypocalcemia. This is due to dysgenesis of the parathyroid glands. Finally, the 22 can help you remember that this is a deletion of chromosome 22q11. This is not DeGeorge syndrome, but is a great example of hypertelorism. Notice that her eyes are very far apart from one another. Okay, let's do a question. A healthy 24-year-old female delivers a male at term. In the delivery room, he is examined and found to have a cleft lip, cleft palate, a cardiac murmur, and hypertelorism. Which of the following is most likely true of this patient's condition? A. He will have an extremely friendly personality. B. He may exhibit a positive Schwastik sign. C. The diagnosis is confirmed with a Western blot. Or D. The condition is due to aberrant development of the 1st and 2nd pharyngeal pouches. Hopefully from the question stem, you notice that this boy has a cleft lip and palate, a cardiac murmur, and hypertelorism. These findings are consistent with DeGeorge syndrome. As we've been discussing, this is due to aberrant development of the 3rd and 4th pharyngeal pouches and can result in hypoparathyroidism. Because the parathyroid glands regulate calcium, he will most likely develop hypocalcemia. The Schwastik sign is a finding seen in hypocalcemia and is elicited by tapping on the facial nerve. If the sign is positive, then this will result in contraction of the facial muscles. So decreased parathyroid gland activity results in decreased calcium, resulting in a positive Schwastik sign. So the answer is B.